evening, ladies and gentlemen. Thank you very much for attending. My name is Andrew Peters. I'm a lecturer here in Indigenous Studies and Tourism at Swinburne University. I'm also a descendant of the Wurundjeri and Yorta Yorta peoples, and it's my great pleasure to be your MC tonight. Before we begin, I'll speak English rather than my native Wurrung language, if that's okay. Before we begin, I'd like to remind everybody to please turn off your mobile phones, or as we sometimes call them, your modern message sticks, or switch them to silent, please, so that we can go through. I'd like to remind you that tonight's presentation, including the questions, will be recorded as a vodcast and will be available to access via the Swinburne website after the event. As I briefly mentioned, I'm a descendant of the Wurundjeri and Yorta Yorta peoples, and one of my great thrills of working for this university is the attachment that we have created around acknowledging our connection to Wurundjeri and Wurrung culture. Part of that is understanding that we live in a very modern world, a world that's dominated by a particular way of thinking that in many views is very different to the way that my ancestors lived their lives on this land today. However, what we do have in common is a relationship, a sense of relationship to each other, and particularly to the land that we're on. For that reason, I think it's really important that in the world today we can maintain ancient connections with the land that we're on. We're in a relatively new building here, the Advanced Technology Centre, and we're very, very proud of the building itself. I believe it's won awards for design and all sorts of things. It's got electronic elevators, so you don't have to walk upstairs anymore. If you're up on the 10th level, you can almost see the ocean, which is where a lot of us go to relieve some stress. But for all that, the foundations of this building are very much rooted in Wurundjeri land. All of the buildings that we occupy in our daily lives today are built on Wurundjeri land. Now that is the essential connection that I'm talking about, that each of us today have a very, very strong connection to the very land that Wurundjeri people walked on for thousands and thousands of years. As part of that culture, before the Europeans arrived, they would often gather for a variety of reasons, one of which included exchanging information. I made a little joke before about the modern message stick being the mobile phone, but indeed message sticks were a very important part of communication for Wurundjeri people. And so a gathering like tonight would have been held in many ways to exchange information and to hear from experts, elders who were wise and were able to share that information with people who were able to listen. So I think it's really appropriate and a wonderful experience for me to be able to perform for you tonight an acknowledgement to country and acknowledge that very connection to Wurundjeri country. And the event that we're holding tonight, the exchange of information that we're all going to be part of, is being held on the land of my ancestors. And so we can hope, and I certainly am very confident, that the evening will run as smoothly as the Wurundjeri elders held their events way back when. So in closing, and with permission from my elders, I'd like to open the event by saying, Wurundjeri Balwa Minjika Yimin Kundibik, the Wurundjeri people welcome you to their land today. Now to talk like a white fella. Welcome Chancellor, Vice-Chancellor, members of Council and Senior Executive, guest speaker Professor Shanthi Rajaratnam, Mr Ian Dicker and members of the Dicker family, distinguished guests, staff, students and friends of Swinburne, to this year's Barbara Dicker Oration. We were all most impressed by the excellent presentations from three of our researchers at the showcase prior to the oration. If you didn't have a chance to see the presentations and you're interested in finding out more about the research that's happening here at Swinburne, particularly in our Brain and Psychological Sciences Research Centre, please take the opportunity to look at the exhibition in the foyer if you haven't already. The researchers will also remain behind after the oration to answer any questions you may have. This vital research into the areas of dementia, depression and sleep disorders wouldn't be possible without the wonderful support of the Barbara Dicker Brain Sciences Foundation, established through the generosity of Ian Dicker and the Dicker family, in honour of Ian's late wife, Barbara Dicker. It is now my privilege and my pleasure to introduce Ian Dicker to say a few words for us.
Thank you, Andrew, and Chancellor and Vice-Chancellor and guests. I add my welcome to you to tonight, and thank you very much for attending. I first want to congratulate Swinburne University for the work that they're doing, have done and are doing. We have been working with the university for 4 years now, and in that time they have commenced 40 individual research projects. You saw 3 tonight. They've finished many of those, but as important as anything is that 4 of those 40 have gone to a second stage with another source of funding, significant funding, so that what seeds we've been able to put into the ground, really some pretty big trees coming out there, and that's congratulations to Swinburne for what you've done. When we discussed it as a family on what we wanted to do, I said to the family that the research that's done probably won't make any difference to me, and it may not make any difference to my children, but our hope was that at the grandchildren level, the community would get the benefit of these research. People have said to me tonight, mightn't have to wait that long, but it'd be good. But that's what we're about, is trying to provide for the community opportunities to improve their health in the areas that, the small areas that we're looking at: depression, dementia and sleep disorders. But the university hasn't only done 40 research programs, they have learned and improved on their way in how we allocate the 10 programs a year. In the last policy, we said there must be some for students and some for senior researchers. That's really good that the students have got opportunities. The university has taught people who are applying for projects how to apply. So that's a learning process on what they're doing. They've also learned how to judge those submissions. So I remember one year there were 29 submissions, so they had to judge which 10 got it. And they've got a very well organised program which they put to the foundation on how they do it. And that's all congratulations to the university. To Barbara, she was beautiful to all. Her children, she was a wonderful mother. Her grandchildren, they loved her. To her friends, she was wonderful. And as a physiotherapist, she always paid more attention to people than just the physical problem they had, and that was Barbara. I'm sure she would be absolutely thrilled with the work that's going on in this area. Barbara, I don't know if Hawthorne's a derivative of the Wurundjeri tribe or not, but she was Hawthorne. She grew up in Hawthorne. Her parents obviously lived in Hawthorne. They went to the Hawthorne Primary School. Her 2 sets of grandparents grew up in Hawthorne, so whenever there were other family additions, she was staying with them, so she had all of her time until into her late teens in Hawthorne, and an important part of that was walking the streets around us here today, the lanes around Swinburne, the streets, and of course the most important walk of all was down to Glenferrie Oval to watch the Mighty Hawks. So Barbara really was a vital part of this area, and so it's such a perfect match that the lady we're honouring and Swinburne and Hawthorne are working together. It is a privilege for our family to be working with Swinburne. We're absolutely thrilled with that privilege, and I'm looking forward, Professor, very much to your help for me to get better sleep.
Thank you very much, and just to clarify, Hawthorne is definitely not a Wurundjeri word. I'm a Richmond supporter, so I take a little bit of pride. That's funny, is it? Trust me. I'm going to ask the Professor later about the effect of your team losing a final on sleep, because I didn't get much Sunday night. Hawthorn is, I believe, a flower or a bush or some type of flowering thing, which is definitely not Wurundjeri. In fact, there is only one AFL team with an Indigenous name. Have a think about that, and if I remember at the end, I'll tell you what it is. But I digress. Thank you very much, Anne. It's a great pleasure, and it's always wonderful to hear you speak, and particularly about your lovely wife in that way. And we're very, very proud, I know, as a university to be associated with your family in this way. I'd now like to invite Graham Goldsmith, the Chancellor of Swinburne University, to open the oration and to present our guest speaker tonight.
Thanks very much, Andrew, and certainly want to echo the words that you said about Ian and Barbara, and thank you for your words about her, about Swinburne and about Hawthorn. I would also like to begin by acknowledging the traditional custodians of the land on which we gather, the Wurundjeri people of the Kulin Nation. We also pay respects to all Aboriginal community elders, past and present, who have resided in the area and have been an integral part of the history of this region. I'd like to extend a very warm welcome to Ian, Beverly and the Dicker family and all your friends, the State Member for Hawthorn and Shadow Attorney General, Mr John Bizzuto, MP, former Chancellor of Swinburne University, Doug Mitchell, my council colleagues and senior executives of the university, alumni, colleagues and friends of Swinburne. And also a very special welcome to our guest speaker this evening, Monash University Professor Shanthi Rajaratnam. And I'll speak a little bit more about him shortly. It is through the generous support of the Dicker family that Swinburne was able to establish Australia's first multidisciplinary mental health institute in 2012. Many of us here tonight have been touched in some way by depression, dementia, sleep disorders or anxiety. And this may have been through personal experience or through a family member, friend, neighbour, or even a work colleague. The Barbara Dicker Brain Sciences Foundation supports applied research into brain and psychological sciences. The ultimate goal is to find a cure, treatment, or prevention for these conditions. The research showcase before tonight's oration highlighted a number of Swinburne projects that are making a difference in the lives of people with mental health conditions. Another wonderful example of our recent work in this area is a project that saw Swinburne's Dr. Michelle Lim lead a team that developed a smartphone app and online intervention program designed to reduce loneliness. This is a great example of how new and emerging technology can make it easier for people to seek help when they need it. And it's research projects like this that make Swinburne incredibly proud and grateful to be affiliated with the Dicker family. It's also been excellent to see Swinburne and Monash work together over the past 5 years to build a strong research network. We value the close partnership that we have with them. It has allowed us to make vital breakthroughs in the area of sleep, neuroscience and mental health. Swinburne has office space embedded in the clinical service area of the Monash Alfred Psychology Research Centre, and these strong clinical links also encompass St Vincent's Hospital. Our guest speaker for tonight's oration is Professor Shanthi Rajaratnam. He is the Deputy Head of the Monash School of Psychological Sciences, leader of the Sleep and Circadian Medicine Laboratory at Monash, and Chair of the Monash Sleep Network. Professor Rajaratnam's postdoctoral training was from the Centre for Chronobiology at the University of Surrey in the UK and the Harvard Medical School in the United States. He's a registered psychologist, chartered psychologist in the UK, and an associate fellow of the British Psychological Society. He is immediate past president of the Australasian Sleep Association and is a member of the Harvard Work Hours Health and Safety Group. He has more than 20 years' experience studying circadian biology and medicine, in particular the circadian regulation of sleep wakefulness. Tonight, the Professor will address a subject that affects our health, safety and productivity every day, sleep loss. Ladies and gentlemen, please join me in welcoming Professor Shanthi Rajaratnam.
Thank you very much.
Well, thank you very much for that kind introduction, and I'd like to also acknowledge the Dicker family, particularly Mr. Ian Dicker, the Barbara Dicker Brain Sciences Foundation, the Chancellor and Vice-Chancellor, distinguished guests, members of the community from Swinburne and elsewhere. It is a real honour and pleasure to give this lecture. I feel thrilled to do it, but I also feel somewhat humbled from all of what I've heard of the work that is going on here at Swinburne and elsewhere. So thank you for the invitation. So I'm going to talk about a topic that is very close to my heart, and that is sleep. And in this short time, I'm going to be talking to you about one aspect of sleep in particular, and that is I'm going to focus on what we call the biological clock, or the circadian clock, which those of you who attended this excellent research showcase earlier would have heard Professor Greg Murray talking about the circadian clock in the brain. So sleep has become a very significant area of inquiry in the health sciences over the past few decades. Why is that? It's because we know that sleep has broad significance for human health, as well as for maintaining optimal safety, productivity as well. Bodies such as the Institute of Medicine of the National Academies in the United States have drawn conclusions based on the evidence concerning the link between sleep loss, sleep disorders, and human health. And they recognise that sleep loss and sleep disorders represent an under-recognised public health problem. They're associated with a wide range of health consequences, including increased risk of high blood pressure, diabetes, obesity, depression, heart attack and stroke. Almost 20% of all serious car crash injuries in the general population are associated with driver sleepiness. Some experts have therefore concluded that makes driver sleepiness the largest identifiable and preventable cause of accidents in all modes of transport, surpassing alcohol and drugs. And so this is a significant public health problem. Now, I want to draw on two examples of the work that I have been doing in collaboration with the Division of Sleep Medicine at Harvard Medical School to illustrate the broad public health significance of sleep loss and sleep disorders. The first study is a study of police officers that lived in the United States, and each of those little blue dots represents a single police officer that we studied, about 5,000 in total. And the first thing we did was that we screened them for common sleep disorders. The sleep disorders included obstructive sleep apnea, which is a highly prevalent sleep disorder that's characterized by obstruction in the upper airway that causes sleep fragmentation and intermittent hypoxia, so a reduction in blood oxygen levels intermittently. Insomnia, which is another very common sleep disorder, which I'm going to be talking about in more detail shortly. Shift work disorder, restless leg syndrome, and narcolepsy. To my surprise, we found that about 40% of the police officers in our sample screened positive for at least one of the 5 sleep disorders we assessed. We then looked at the associations between these sleep disorders and a number of health, productivity, performance, and safety outcomes. And so initially, we looked at the relationship between those who had a sleep disorder, such as obstructive sleep apnea, and major health conditions such as depression and diabetes. And we also looked at well-being, we looked at burnout, particularly emotional exhaustion, and the risk of falling asleep while driving. All of the risk of all of these things was approximately doubled, and in some cases more than doubled, when the individual had obstructive sleep apnea, even after correcting for a number of other factors that can contribute to the risk of these particular outcomes. We then followed up these police officers for about 2 years. Now, when we started the study, I was told no police officer is going to admit to making administrative errors and falling asleep while driving. And perhaps we had one outcome was also shouting at citizens as well. But indeed, we did get quite a few reporting these outcomes, and when we followed up prospectively, about 15,000 person months, we found significant increases in these adverse performance outcomes in those who had obstructive sleep apnea. Another example is a study we've just completed and published this year, and again in collaboration with the Division of Sleep Medicine at Harvard Medical School. Here what we did was I worked with a group of legislators as well as the Massachusetts Registry of Motor Vehicles to tackle a problem that had been documented over a number of years in the state of Massachusetts and worldwide, and that is the problem of increased risk of motor vehicle crashes in young people, in 16 and 17-year-old drivers. And one of the factors that's contributing to this risk is the increased propensity to fall asleep at night, the greater vulnerability of young people to fall asleep at night while they're driving. Now, this is one of the contributing factors. What we did was we worked with a group of people to introduce or develop model legislation that included stringent penalties for violating a nighttime driving restriction. There were several other aspects to the legislation as well, but one key aspect was very tough penalties for violating a nighttime driving restriction, so young people aged 16 and 17 could not operate a motor vehicle overnight. And they were deemed unlicensed if they were operating a motor vehicle without a legal guardian or parent in the vehicle. We found, after following up, we looked at motor vehicle crashes over a period of 5 years after implementation of this new legislation. And what we found in the 16 and 17-year-old drivers using a time series analysis is that there was about a 20% reduction in motor vehicle crashes in these young people. Over the 8 years of our study, that would translate to about 320 less accidents that caused fatal or incapacitating injury, and about 13,000 less accidents involving these young people. So this was a major policy intervention that resulted in significant reductions in motor vehicle crashes and tackled one of the manifestations of sleep loss and sleep disorders in the population. Now, sleep must be important. We have, over the course of years of evolution, conserved this function. We spend about a third of our lives sleeping, and that means if we are lucky to live to the age of 90, about 30 years spent sleeping. But it's only in recent years have we begun to understand what some of the vital functions that sleep might play. One area that was an area of significant interest for cognitive neuroscientists in particular is that sleep is an active state, and during sleep, memories of the day are consolidated. And here is an example of an experiment that showed this very nicely in Bob Stickgold's group at Harvard Medical School. So participants were brought into a laboratory. They were taught a pattern recognition task. And after the first night, they were, immediately upon exposure to the task, they were tested. And then the next day and subsequent 7 days, they were tested on this task. And the conclusion is that over several days, these individuals showed significant improvement on the task. But if on the night after learning the task, they were deprived of sleep, and then they were given a couple of nights to recover, so they went to sleep on the subsequent nights, and then they were tested on day 3, there was only a very modest increase in improvement in memory or performance on this task. And this and other experiments like it provide a strong evidence base for the role of sleep in memory consolidation. Recently, the role of sleep in flushing out toxins has been described, particularly in animal models. And this was a study published in Science a few years ago showing that an area of the brain, or a network in the brain called the glymphatic system, is significantly altered during sleep. This plumbing system in the brain, it's thought during sleep, the space between these, or the space occupied by this system increases to allow more fluid to pass through it. And what these researchers were able to demonstrate is not only was there about a 60% increase in the space occupied by this system, but then if they injected toxins into the brain or into the cerebrospinal fluid of mice, the toxins were more rapidly eliminated during sleep or when the mouse was anaesthetized compared to when the mouse was awake. And so in this particular graph, what you're seeing is labeled beta amyloid, a protein that is involved or associated with Alzheimer's disease. And when this protein was injected, you can see that the rate of elimination of the protein is more rapid when the mice are either asleep or anaesthetized compared to when they're awake. And this is, again, a very active line of inquiry now that sleep plays this role in eliminating toxins. Now, the problem of insomnia is growing in our society. And when we look at GP data, or audit data, we can see that insomnia presentations are very common in primary care. We also are when we look at the way in which insomnia is currently managed in primary care, perhaps we despair a little, because we look at, firstly, about 95% of insomnia presentations in general practice are managed with prescribed medications, largely benzodiazepines. And we also, compared to about 54% of all presentations in general practice. We also see that despite this country having significant expertise in sleep medicine, we have about 0.8% of insomnia presentations in general practice are referred on, compared to about 8% of all presentations in general practice. The GPs are not referring sleep cases on, and there are several reasons for this that perhaps if we have time afterwards we can talk about. Now, there is an effective psychological treatment for the management of insomnia, and this is well established, and here is a meta-analysis led by David Cunnington's group that I collaborated with them in the development of this study. And it's a psychological treatment called cognitive behavioral therapy for insomnia. It includes addressing the cognitive aspects of insomnia. Stimulus control, sleep restriction, sleep hygiene, and relaxation are core components of this cognitive behavioral therapy program. And it was shown in a meta-analysis, bringing together all of the evidence concerning the efficacy of this treatment, to reduce key indicators of sleep quality. That is, how long it took you to fall asleep, sleep latency, how much you woke up after you fell asleep, the total sleep time, and how efficient or consolidated your sleep was. And those results are comparable to the effects of hypnotics. That is, that cognitive behavioral therapy is as effective as hypnotic medications and probably more effective in long-term management of insomnia. Now, one of the particular interests of our group, and also the group here at Swinburne, you would have seen from Professor Murray's presentation earlier, is the interactions between mood disturbances and insomnia. Previously, it was thought that insomnia was a symptom of depression. And now there is increasing evidence that insomnia may actually occur prior to the depressive episode and may actually precipitate the depressive episode, and there is this complex interrelationship between the two. And so one demonstration of that is a study that a DPsych student of ours conducted a few years ago, a randomized controlled trial where he took cognitive behavioral therapy for insomnia that I've been just talking about and gave it to patients that had major depression. And these were patients with major depression who were taking antidepressant medications, but for whom their depressive symptoms continued and their insomnia also continued. And so, as expected, over the weeks of the study, those who received the cognitive behavioral therapy for insomnia compared to self-help therapy showed a marked reduction in insomnia symptoms. But what we also saw is this marked reduction in depression symptoms. That is, the therapy targeted the insomnia, but there was a reduction in depression. And this is also compelling evidence that insomnia can actually play a causal role in the development of depression. Now, we're taking this work further to try and tailor cognitive behavioural therapy for circadian rhythm disturbances, because circadian rhythm, body clock disturbances, are one cause of sleep-wake disturbance that may manifest as insomnia. You may have heard of something called chronotype. And chronotype, if I was to give you all a questionnaire called a morningness, eveningness questionnaire, you will all fall somewhere in a distribution of ranging from a lark, an early morning type person, to an owl. And maybe I don't even need to give you a questionnaire. I could probably ask you and you'd probably know. So if people fall in this spectrum, there is a normal distribution, and we know that the extreme owls and larks, there are genetic differences between these populations as well. We know also that if you take a group of people that have major depression and give them the cognitive behavioural therapy for insomnia that I was talking about, those who are more evening type tend to benefit less from the treatment. So that is that there are some differences. Even though on the whole cognitive behavioural therapy for insomnia is effective, there are subpopulations that appear to benefit less from the treatment. This was research done by Dr. Bay, who's now at Monash in collaboration with the group at Stanford. So naturally, this is our interest in teasing apart these potential contributory factors to the ongoing sleep-wake disturbances, like in Dr. Bay's study that I talked about. So we are particularly interested in the regulation of the sleep and wake cycle, in particular the contribution of the circadian or 24-hour clock. There is also another mechanism that is known to drive the sleep-wake cycle, the so-called sleep homeostat, which is more like an hourglass that flips over every time you wake up. It continues to accrue sound, and it keeps timing, or it keeps the duration of your wakefulness, and it maps that, and it increases your sleep load the longer you stay awake. And if you deprive yourself of sleep, you fall asleep easily because of this homeostat. Of course, we humans are terribly social, and that significantly impacts the sleep-wake cycle. And we know the light and dark cycle is the major regulator of the circadian timing system, and I'm going to be talking about light in a moment. So just very briefly, the master circadian clock in the brain is located in an area of the brain called the hypothalamus. It's referred to as the master circadian clock. Its role is like the conductor of an orchestra. Its role is to regulate timing within the brain and throughout the body of physiological and behavioural events, and maintain this 24-hour time course of several processes to make sure that the timing is appropriate for the internal organisation within the organism, as well as our relationship with the outside environment. So the circadian timing system plays a crucial role in making sure the light and dark cycle and our physiological processes are appropriately synchronized. And we see several examples of circadian rhythms, and this was a paper we published several years ago showing the melatonin rhythm, which I'm going to be talking about more in a moment. The melatonin rhythm melatonin is a hormone that is synthesized and secreted by the pineal gland in the brain. It is involved in the regulation of our sleep-wake cycle, and it's widely used as a marker of the circadian clock. How do we tell what time it is according to the clock? Well, many of us have started to use the melatonin rhythm as a signal, if you like. It's like peering into the brain clock through using the melatonin rhythm. And there are several other rhythms that are illustrated there. Now, what I'm going to show you is a series of studies we've just completed looking at the significance of the timing of the body clock, or the circadian clock, for our cognitive function, our cognitive health, as well as our mental health. And the typical way in which we assess the timing of the circadian clock, as I mentioned, is through the melatonin rhythm. So if you just look at this graph here on the top right-hand side, what we have is the onset of the evening melatonin secretion is plotted relative to when you go to sleep. That is, and each of these circles represents one participant in the study. So you could see the wide inter-individual differences that we note in this population of individuals we studied. On average, and the triangle shows the average, melatonin starts to be secreted about 2 hours before we go to sleep, and that's the normal phase relationship between the melatonin rhythm and our sleep. But we see several individuals where the timing is much earlier. Melatonin starts to be secreted several hours before, or even in some cases after the individual goes to sleep, and this is what we would call inappropriate timing or misalignment of the circadian system from the sleep-wake cycle. If we take all of these individuals and expose them to a couple of nights of inadequate sleep, which I'm sure many of you might be exposed to anyway, and then we assess their cognitive function the following day, what we find is those who have a tendency to have a short, for their melatonin rhythm to be occurring close to the time of their sleep time or afterwards, those tend to have more lapses of attention. They're more impaired compared to those that have the appropriate timing of the melatonin rhythm. That is, circadian rhythm misalignment is associated with impaired cognitive function. We also see relationships between the timing of the melatonin rhythm and several mood parameters that we assess. These were healthy individuals that have these clear relationships. Now, in the mental health area, this relationship has been well documented in a disorder known as seasonal affective disorder, winter blues. Thankfully, in Melbourne, this is not a phenomenon that is widely reported, or there are not a high number of presentations, but we see in the Northern Hemisphere a significant problem with winter depression. And one of the hypotheses for why in winter there is this type of depression that manifests is the relationship, the alteration in the relationship between the circadian system, the sleep-wake cycle, and the light and dark cues that occur during the winter months. And so the typical relationship is that we see a melatonin rhythm, as I said, that precedes sleep by about 2 hours. The onset of melatonin is about 2 hours earlier than the sleep onset. But what we see in a group of patients with seasonal affective disorder is that as their melatonin rhythm becomes misaligned from the sleep-wake cycle, so if we look at these extreme ends of the curves, there is increased depressive symptoms. There is a clear relationship between depression in this population of seasonal affective disorder patients and misalignment of the body clock. Delayed sleep phase disorder is a very common disorder in adolescents particularly. It is the disorder which many of you may have yourselves experienced, or have teenage children who you experience the difficulties waking them up in the morning. Well, this is the disorder whereby the circadian system shows this rapid delay in the early adolescent years, such that sleep becomes much later, and it's very difficult to wake these individuals in time for school or work. As a result, in the Northern Hemisphere, there have been calls for shifting schools to a later start time to accommodate the fact that biologically these individuals are not well adapted to be learning productively in the morning hours when their clock is really signaling sleep. When we look at people with delayed sleep phase disorder, an extreme version of this kind of delay in the sleep-wake cycle, we see significantly impaired productivity outcomes, high levels of absenteeism and reduced productivity, disrupted work and school, social activities, family, home responsibilities in these adolescents that have delayed sleep phase disorder. We've just completed a study. Jade Murray is a PhD student in our group, where we've examined DSPD, or delayed sleep phase disorder patients. We brought them into the laboratory, so we got a sleep physician to categorize these patients as delayed sleep phase disorder. And so we screened them very carefully, had them formally diagnosed by a physician, and then we assessed several outcomes, including depressive symptoms and illness severity. And what we looked at is we looked at some of the patients, we found that when we assessed their melatonin rhythm, they had a true delay in the melatonin rhythm. And we called these ones the circadian group. And then when we assessed the rest of them, they didn't really have an internal delay or their melatonin rhythm was not abnormally timed. Their sleep pattern was probably being driven, their poor sleep was being driven by causes other than a delayed circadian system. But when we compared these two groups for important outcomes like depression and also the overall illness severity rated by the clinician, who didn't know which group the patient belonged to, we see significant differences between the two groups. The work has also been extended. This is a collaboration between our group and the Brain and Mind Research Institute in Sydney, looking at patients with mild cognitive impairment. And there are clear associations between the timing of the melatonin rhythm and the severity of the cognitive impairment. In fact, earlier dim light melatonin onset, that is the timing of melatonin, was moderately associated with poorer consolidation of verbal material in the evening. And there was no such association in healthy controls, and there was earlier melatonin timing in those with mild cognitive impairment. Here is work by Greg Murray done here at Swinburne that also corroborates this line of investigation whereby you see an association between circadian rhythm disruption and, in this case, vulnerability to bipolar disorder. And this was a study in which Greg looked at using actigraphic, so wrist actigraphy, activity rest cycles measured using a classic device. Many of you are wearing something like it. I've got a Fitbit on here, but there are many, many devices like it that have an accelerometer. Well, in sleep research, we use an actigraph that's similar to this, and we have the ability to then continuously record activity rest cycles. And using the amplitude or the level or intensity of activity, Greg was able to classify these patients into different groups and look at their the relationship between the amplitude of their activity rhythm and their propensity to have bipolar disorder, and there's a clear relationship. Now, I've been talking about cognitive health and mental health so far, but I just want to very briefly touch upon the broader implications of circadian rhythm sleep disturbance. So this was work published by Frank Shear and colleagues several years ago in the Proceedings of the National Academy of Sciences, and what they showed was that disruption of the sleep-wake cycle from the light and dark environment results in disruption of the internal organization within the body, and this can result in disruption of the fasting feeding cycle and also metabolic cycles, resulting in metabolic dysfunction. What they did in this study is they brought healthy young people into a sleep laboratory, and they measured a hormone called leptin, which is associated with our appetite. Higher leptin levels indicate increased satiety, so reduced appetite. And what they did was they measured in this group of individuals leptin levels across the day while people were awake and asleep. And then what they did was they progressively shifted their sleep by 4 hours each day, so that the body clock, the circadian clock, became misaligned from the sleep-wake cycle. And they did this over several days in the study, in the laboratory environment, and they continued to measure leptin levels. And they found that leptin became increasingly suppressed as sleep became misaligned from the body clock. And the conclusion from this study is that this may contribute to be a mechanism underlying the increased risk of obesity among shift workers, for example, who are continuously challenging the sleep-wake cycle and the body clock. They also found in this group of healthy individuals, they measured such things as their glucose regulation. And I won't go into detail, I don't have time, but they found that in the misaligned state, 3 individuals who were perfectly healthy before were entering the pre-diabetic state when they were in a misaligned phase. Thankfully, it was reversed when their sleep came back to the normal time. But again, there is a strong, as I mentioned earlier, an association between sleep-wake disturbances, diabetes and obesity, and these laboratory studies are providing insights into possible mechanisms. Finally, I want to just conclude with talking about the work that we're doing in terms of the interventions that we're using. And for circadian rhythm sleep disturbances, I strongly believe that we should use therapeutic agents that target the circadian timing system. So if we look at the key parts of the circadian timing system, the suprachiasmatic nuclei, the circadian clock of the hypothalamus, the retina, which brings light information to the hypothalamus, and this multisynaptic pathway, the pineal gland, which produces melatonin, which feeds back and regulates the circadian pacemaker. And so we've done work looking at the role of melatonin and melatonin agonists in the treatment of circadian rhythm sleep disorders. And here is some work that we published a few years ago, testing a drug that acts on melatonin receptors. And the figure is complicated, but just imagine that you're brought into a sleep laboratory and you live there for a week, and we simulate a flight, and we did the study in Boston, so we simulated a flight from Boston to London. Sadly, when I told these participants this, they got quite excited about simulation from going to Boston to London, but the only thing that really changed was their sleep times. There was nothing else that changed. They didn't go to London. And so in the laboratory, we took them to London. We brought their sleep-wake times 5 hours earlier than it would have been. So what you see here is the time of day, and the colour represents the intensity of sleep. Red represents very intense sleep, and according to this colour spectrum. And you can see on the first day, the baseline day, where they slept at the local time zone, sleep is very good. It's very consolidated. Then when sleep was shifted to 5 hours earlier for 3 nights, you can see that it became very disrupted. But when we gave increasing doses of a melatonin agonist, we were able to restore sleep to or to baseline levels, particularly in the highest dose. So melatonin agonist tasimelteon was able to improve sleep quality in this kind of jet lag simulation, a model of circadian rhythm sleep-wake disturbance. Recently, Steve Lockley, a colleague of ours who has a part-time appointment now at Monash, published a follow-on study of this same melatonin agonist, tasimelteon, 20 milligram, in a population who have profound sleep-wake disturbances, and that is individuals who are totally blind and lack photic input or lack light input. Therefore, their circadian rhythms free run. They exhibit their internal periodicity. They fail to synchronize to the external environment because the main time cue is lacking. So he took a group of patients that have this disorder called non-24-hour disorder, and then studied firstly their sleep and wake cycle over several days, and you can see they try and maintain a 24-hour cycle, but these little stars represent the endogenous melatonin rhythm, the timing of the circadian pacemaker, which, as you can see, is drifting all over the place, right? It's free running, it's shifting along. If it was synchronized, it should be a straight line. And when they were treated with tasimelteon, indeed, the circadian system aligned. And in the placebo condition, there was no such alignment. And so hence, this melatonin agonist is now proposed to be used, and indeed, it has been approved by the Food and Drug Administration now for the treatment of non-24-hour disorder in the blind. The work, and I just put this up to remind us of the contribution of Australian science to these global developments. This was a study that was done by Jenny Redman, who happened to have been my PhD supervisor, more than 30 years ago, where she tested this in a rat model at La Trobe University, and she published in Science what was a groundbreaking finding, that is that melatonin can actually synchronize the activity rhythm of the rat. And this work was exactly the same protocol that was ultimately used by this pharmaceutical company that then were able to get FDA approval for the melatonin agonist. The second and final area that I'll conclude with is light, and we're using light as an important time cue now. We're using light treatment for circadian rhythm sleep disturbances in a variety of neuropsychiatric disorders. And just to illustrate the growth in understanding of the neurobiology, the mechanisms through which light affects the central circadian clock, we know now that there is a dedicated circadian photoreceptor mechanism that is maximally sensitive to short wavelength or blue light, and therefore devices, lighting systems, including architectural lighting systems, are being designed to make use of these important advances in our understanding of how biologically we respond to light. Our circadian system responds maximally to blue light. And so we've been doing some work understanding the time course of these effects and comparing these, for example, to green light, which we know that classical photoreceptors are maximally sensitive to. And we've been taking these findings into clinical situations. So, for example, here is a colleague of mine that I work closely with, Professor Jenny Ponsford, who is a clinical neuropsychologist and a doctoral student who conducted the work. What we did was we used a blue light device compared to a device where we depleted or filtered out the blue, and we did a randomized controlled trial in a group of patients that had suffered a traumatic brain injury and who show significant fatigue and sleep disturbance and mood disturbance as a result of the injury. And we found that compared to the yellow light or treatment as usual, or no treatment, we found that the blue light significantly reduced daily exposure to the blue light, significantly reduced fatigue and sleepiness, and we've just conducted an extension of this particular study funded by the Victorian Neurotrauma Initiative. And so I talked about the fact that architectural lighting has enormous potential to influence the way in which we manage certain neuropsychological and psychiatric disorders. Here is just one example from a group that published a few years ago a light intervention in an aged care facility to reduce dementia symptoms. And so what they did was they increased light intensity in this aged care facility, and they were able to show that the light attenuated cognitive deterioration, it also ameliorated depressive symptoms, and it attenuated the increase in functional limitations over the course of the study in those who were exposed to the brighter light. And so now that we understand the colours, the wavelength of light that's important, we are doing quite a lot more than this. So just in conclusion and future directions, I hope I've convinced you that sleep loss, sleep disorders and circadian misalignment adversely affect cognitive function, productivity, safety and health. Sleep disturbances and circadian rhythm misalignment are highly prevalent in certain neuropsychiatric disorders with possible causal interactions. The role of the circadian system in maintaining optimal health and well-being is being increasingly recognised. And circadian phenotyping, that is characterising our individual body clock, and tailored treatment approaches have significant potential to improve health outcomes. What are we doing about it? Well, we're a part of, in fact, Monash leads a cooperative research centre in this area, the Alertness Cooperative Research Centre, which is developing products and tools to try and manage impaired alertness, safety and productivity, particularly in occupational and transport settings. So one of the areas that I'm actively involved in this centre is working with Philips, which is the major end-user participant in the Cooperative Research Centre, to develop a diagnostic test to characterise our circadian timing system. So I was thrilled to see the work that Greg's group is doing here in Swinburne, which is developing a questionnaire to try and disentangle mood, sleep-wake disturbance and circadian function, funded by the Barbara Dicker Foundation, because this will be a very nice adjunct or parallel to the work that we're doing, which is developing a blood-based diagnostic test. And in order to recognise and support the clinical training and the research in this area, we've established a clinic. And it's a clinic that is a multidisciplinary clinic that specialises in insomnia and body clock disturbance, particularly in shift work and in delayed sleep phase disorder. So finally, I'd like to thank all of the collaborators that have contributed to the work, particularly those in the School of Psychological Sciences, the Division of Sleep Medicine, the University of Surrey, and several other institutions that we have active collaborations with that have contributed to the work that I presented here. All of the funding agencies, including the Alertness Cooperative Research Centre and the Neurosleep NHMRC CRE, which we're proud to be a part of. I'd also like to acknowledge my family, who continue to provide ongoing support and encouragement, and especially my parents, who made the decision to come to this wonderful country and give their children opportunities. Thank you.
Thank you, Professor Rajaratnam. We now have just a few minutes for some questions. If there are any laymen in the audience like myself, a lot of them will revolve around what the words meant that you were talking about. But I'm sure we have a number of experts who'd be happy to ask the Professor some questions. We have some roving microphones.
I'm always fascinated by this fact that if you deprive people of light, they go into a 25-hour clock. What does that mean?
Well, if you deprive, and Jenny Redmond's experiment that I showed you earlier is a classic illustration of that in a rat model, and in humans respond similarly. So if you take away the light and dark cycle, our circadian clock in the brain begins to express its own natural periodicity. That is, we have evolved to be organisms that have a day length that actually deviates slightly from 24 hours. Now that has puzzled many, and there have been many explanations for why that is. The majority of people have a clock that's slightly longer than 24 hours. You mentioned 25, that came from an early study that was done in Germany about 50 years ago, and the more recent evidence is that it's actually closer to 24. We think it might be because having a clock that deviates slightly from 24 hours allows for adjustment more easily when there is an environment, a change in the environment. Now, my physicist colleagues that model the circadian timing system tell me this, that if you have an oscillator that deviates slightly from the 24-hour cycle, it needs to be corrected each day, and when there are changes in the environment, like fluctuations in day length, this produces a more stable system and it's able to synchronize more easily to the changes in the environment. That is one theory that has been put forward.
I was interested to hear you talk about your family, Professor. I had visions of you watching them sleep night after night, caring your children.
I have, and just perhaps if I can mention that, I often in lectures talk about the fact that having children taught me as much as I could possibly learn about sleep and sleep deprivation. And one of the things I talk about in the lectures, if you watch a young child sleep, you can see sleep architecture beautifully illustrated because they have such strong homeostatic sleep pressure. They go to sleep, hopefully, on time, and they then are in deep sleep. And we'd have like dinner parties and things where people would come around, and this is when the kids were young, and they'd say, well, why aren't you going to wake up the children? I'd say, no, they would be somewhere in slow wave sleep at the moment. There's no chance, but you've got to get out before 11 o'clock because they'll be starting to arouse around that time.
So my question has to do with slightly older children, namely the whole issue of screen time and teenagers and others looking at the screens of their smartphones and the like before they go to sleep. Now, there is the activity of actually texting or whatever, but what about the issues such as the lux or the light that they are experiencing? Does the research suggest that that is an additional factor to sleep misalignment and problems with sleep hygiene? So
it's a very active area of study. In fact, we have an NHMRC-funded trial going on at the moment looking at the impact of lighting on the development of delayed sleep phase disorder. So what we think is happening is that some individuals have a propensity to develop delayed sleep phase disorder, that exposure to light in the evening, which is the time of day that the circadian system is sensitized to light in such a way that it will cause the clock to move to a later time. So this thing, what the exposure to light is making the manifestation of delayed sleep phase disorder much worse. So there is evidence, David, in studies in healthy individuals that has shown that the light levels that can be emitted by a backlit device are sufficient to suppress synthesis of melatonin and delay the timing of melatonin rhythm, which suggests the circadian system is affected, and it can also affect sleep quality. It's one of the most common questions that I'm asked is, so then what should we do with iPads and these devices? Well, at this stage, until we understand more broadly the implications of these devices, I say we'll certainly switch them off as close as possible to sleep time. Secondly, there are apps available that reduce not only the intensity of light, but the spectral composition of the light. I was saying earlier that the circadian system is most sensitive to blue light. There are apps available that reduce the blue content from the devices in the late evening hours to reduce the circadian response.
I was wondering whether your clinic deals with primary school children who have sleep difficulties. You were talking about adolescents and adults, but I wasn't sure whether you also included younger children in your studies there. Yes?
So in our research studies, I've typically worked in adults with sleep-wake disturbances, but my colleagues and our clinic at the Monash University's Healthy Sleep Clinic is primarily dealing with adults. But as a part of the Monash Sleep Network, one of the clinical centres that are affiliated with Monash is the Melbourne Children's Sleep Unit, and there are excellent paediatric sleep physicians there, and they run an excellent program, as there are in a few other hospitals as well. But
okay, thank you very much, Professor. Unfortunately, we've run out of time tonight. I know I've received some information tonight. I've now got a reason why some of my students might fall asleep in my lectures. I'd now like to, it's my great pleasure to invite our Vice-Chancellor, Professor Linda Christensen, to the stage and close the evening for us, even if she is a Hawthorn supporter.
thank you.
Thank you very much. Professor Rajaratnam, on behalf of everyone here, I'd like to thank you for sharing your vital research with us tonight. And in lieu of a speaker's gift, the university is proud to make a donation to the 1509 Friends of Eye India project, which is your request.
Thank you very much. Thank you.
Tonight is a wonderful example of what can be achieved when universities share their expertise in areas of research. Swinburne's strong research relationship with Monash in the area of mental health and neuroscience is helping improve the lives of people around Australia and in the world. And it was wonderful to see your maps and see the connections and the way you're reaching across to find the best minds to help solve these complex problems. I did notice one of your first diagrams or maps showed the police study. And you even had Winnipeg, where I'm from. And my father has seasonal affective disorder. Oh. It's the coldest city on a planet with a population over 600,000. Cold, long winters. So we moved here for a reason. I would like to say another warm thank you to Ian Dicker and his family. Your financial contribution continues to provide Swinburne with the opportunity to generate new knowledge and to contribute to research that has a genuine impact on people's lives. And to everyone here in the audience, if you've seen or heard something tonight that has inspired you, we would love to talk more with you. Our staff are available to chat with you now at the registration desk in the foyer and can provide you with information about the work that we are doing through the Barbara Dicker Brain Science Foundation. Every donation made to the foundation will allow us to increase our research capacity, which ultimately makes a positive impact on people's lives. So to finish, I would like to thank everyone, friends, staff, and students for attending this evening, and we look forward to seeing you at next year's Barbara Dicker Oration. Thank you, and good evening.
This has been a Swinburne production.